Good morning. In fact, good afternoon here.
Yeah.
I'd like to say some words about Professor Emmott. Thank you very much, Professor, for joining us today. And before I start our talk, I'd like to highlight some achievements of your scientific journey. Ed moved to the University of Liverpool in November in 2019 to set up his lab as part of the Center of Proteomics Research Department of Biochemistry. The lab goal is the use and development of proteomic methods to study RNA virus replication and virus host interaction, and particularly the use of single-cell proteomics to aid in this research. Previously, Ed was a postdoc in Nikolai Slavov's lab at the Northeast University, and among the studies he performed during his postdoctoral research, he developed single-cell proteomic methods such as SCOPE-MET and SCOPE2. Prior to joining Nikolai's lab, Ed was a postdoc in Goodfellow lab at the University of Cambridge and Imperial College London. And his PhD was at the University of Leeds under supervision during his course with the thesis on high-throughput proteomics analysis of the interaction of avian coronavirus and nucleocapsid with the host cell. And his bachelor was from the University of Warwick in medical microbiology and virology. Thank you very much for joining today and share your experience. Thank you.
Thank you very much for the invitation. It's a real pleasure to be here. So, okay, hoping that my screen is cooperating now. So,
yes.
So my lab is we use proteomics, dominantly mass spectrometry based, to study viral proteases, and we do this both in normal bulk samples, and we're also interested in studying this at the single cell level. And my talk today is going to have two parts. We're going to talk through a study we did studying viral protease activity at bulk level, and then some of the work we're doing now, some of our ongoing work on applying single cell proteomics to study viral proteases. So why do we care about viral proteases? Well, here is a virus that I suspect everyone here is far too familiar with. The bits people usually talk about are the spike protein. This is what's in our vaccines. Mutations in spike are what tend to drive new variants emerging. I'm more interested in this end of that virus. We have two long polyproteins that are produced, a short one, PP1A, and a longer form that is produced by ribosomal frame shifting. So it has a C-terminal extension. And these are cleaved into between 11 and 16 domains by two viral proteases, NSP3, or the papain-like protease, and NSP5, or the main protease. Now, this seems like quite a convoluted way to express the proteins a virus needs for its replication. So why do coronaviruses do this? And the reason is that cleavage does not go to completion. We don't just see the immediate processing of these polyproteins down into these different fully cleaved domains. Instead, we see the accumulation of cleavage intermediates, and these can have their own distinct roles in virus replication. Early on in infection, you will tend to see accumulation of longer forms of these proteins, and later on you'll see accumulation of shorter forms of these proteins. And the switch from long to shorter versions of these proteins can drive what the virus is trying to do in terms of RNA synthesis at different times of post-infection. So it's a way for the virus to regulate its replication. And it is all driven by the two viral proteases. Now, this isn't just seen in coronaviruses. We can see it in alpha viruses like Simliki forest, in noroviruses, picornaviruses such as polio, or at the more extreme complexity end in the coronaviruses. So this is a very widespread mechanism. So really, the takeaway I want you to have from this introduction is that proteases and proteolysis plays a crucial role in the replication of many RNA viruses, And for this first part of the talk, I'm really going to be focusing on SARS-CoV-2. Now, from very early on in the pandemic, there's been a real interest in these proteases, so the main protease, the papain-like protease, as well as the role of cellular proteases in this viral life cycle. So we wanted to study proteolysis during SARS-CoV-2 infection. Now, for any of the audience who are familiar with studying protease biology, you may be familiar with techniques that come under the umbrella and thermonomics. And this is what we applied. So how do we study proteolysis with a bottom-up proteomic approach? So here we have a hypothetical protein X. It has an N-terminus that's acetylated, as well as a new N-terminus here generated by proteolytic cleavage. And this is what we actually care about looking for. However, during our sample preparation, we're also going to introduce new cleavage sites, which are new N-termini along this protein. So we have a few issues with this process. Our protein N-termini include our native N-terminus, and this could be acetylated, it might have N-terminal methionine or signal peptide removal, and it will also include neo-N-termini generated by protease cleavage. If we can identify a peptide containing this N-terminus, it can tell us both what is protein X, but also what is the location of that cleavage site. So we can get multiple different pieces of information from this experiment. But if we're using trypsin or another enzyme, we're going to generate further new N-termini. So we need a means to distinguish those that are generated by trypsin during our sample processing from those that are actually biologically interesting for us. So how can we study those particular N-terminal peptides? So for any of the audience who are commonly studying post-translational modifications, you're going to be aware that if you really want to study a post-translational modification, you will tend to be needing to enrich. The way we use this, the way we perform this is a modification of the Hunter protocol that was published in Molecular Cellular Proteomics. So you take your samples of interest, this was 2 different cell lines that were infected or mock infected with SARS-CoV-2, and we harvested these at different time points. We lysed these cells, and then in a modification to the protocol, we blocked our N-termini and lysine residues by labeling at the protein level with TMT reagents, so using TMT-Pro. And we were then able to pool all these different time points into a single sample. That sample could then be digested. We then produce new N-termini in this process, which we can label with undecanal. This means when we put this through a C18 column, the undecanal labeled peptides will be retained on that column, and our peptides of interest that the TMT label will flow through. And this gives us a sample enriched for TMT locked N-termini. And we can then further fractionate for deeper coverage by reverse phase. So what did we do? So I've talked to you briefly through our experiment. We either took A549ACE2 cells, so a human cell line, or Vero E6 cells. We infected these with SARS-CoV-2 and harvested at different time points from immediately after infection, early on in infection, to quite late times post-infection. Now, when we looked at our enriched N-termini, we first took a quite simplistic look. If we just look at the very last time point, our 24-hour infected over mock samples, what can we see? So on both of these, we have our 2 cell lines, and we have our N-termini, either viral, cellular, or those N-termini which didn't show a significant change. So you can see in both cell lines, in this upper right quadrant, we see all our viral peptides. This is expected. We'd expect these to be more abundant in our infected samples, but we do also see an increase in cellular N-termini during infection, as well as a large number that also show decreases. So we're seeing changes in proteolysis on infection. Now, I'm first going to talk you through some of our findings with these viral N-terminal. So what did we see? So I'm going to come back to this image here. So we know that the polyprotein is cleaved at multiple sites. We also know that the N protein is quite labile, and you can readily detect cleavage products of N during infection. But what most people are interested in is spike protein cleavage, as this is essential for cell entry. And we were particularly interested because we found a couple of cleavage sites, so one at position 637 and one at position 260. The 637 site in both cell lines we looked for, and it's on this flexible loop near to the furin cleavage site. Now, we wanted to know what these cleavage sites were actually playing a role in. So what could be cleaving these sites and generating them? So candidates would be the viral proteases. Now, for the viral proteases, we have a very good idea of the cleavage sites that these enzymes recognize, and they didn't match these sites in spike. A different possibility was then cellular proteases, and we already know that spike can potentially be cleaved by cathepsin as well as by TIMPus. So how were we able to test this? So we did a second experiment. We took our Vero cells, we infected them with SARS-CoV-2 and allowed infection to proceed for 12 hours. At that point, we added protease inhibitors and allowed infection to proceed for a further 12 hours. Now, if added at the start of infection, protease inhibitors would just stop the virus from growing. We didn't want to do that. We wanted it to be able to express its protein and then simply block this cleavage. And we tested several different protease inhibitors. We tested camostat, which can cleave trypsin, and which can block cleavage by trypsin and TIMPRIS2, and calpeptin, which can block calpain and cathepsin cleavage. And what we found is in our infected cells where we treated with calpeptin, so blocking cathepsin cleavage, we saw decreased abundance of these two sites in spike, and actually of one site in N protein. We didn't see an impact of camostat in this cell line. Now, this was particularly interesting as another group subsequently showed that these are indeed the site of cathepsin L cleavage in spike. So our study was able to identify the cathepsin cleavage sites in SARS-CoV-2. We didn't just see viral proteins cleave though. So I've mentioned the two viral proteases, the main protease MPROT and the papain-like protease NSP3. We have a good idea from prior studies of what sequences these proteases recognize. So MPRO recognizes cleavage sites that look like APSTV, XLQ, and the papain-like protease recognizes this LXGG motif. So when we looked in our data set, we found subsets of new N-termini that matched cleavage at these different sites. So where we have coloring in magenta, this matches the consensus for the MPRO protease, and in green by the papain-like protease. And we saw multiple substrates that showed temporal regulation during infection. So here we have our 0-hour mock, and then we have triplicates for 0-hour, 6, 12, 24, and 24-hour mock. So we can see most of our substrates are showing a strong increase in abundance 24 hours post-infection in our infected and not in our mock cells. A subset of substrates, we can see some evidence for cleavage earlier on, and it tends to be these substrates that actually map the match to the papain-like protease consensus site. So this was interesting for us. None of these had previously been identified as substrates of coronavirus proteases. But it begs the question, the virus is cleaving cellular proteins. Why is it doing this, and is it actually important? So why might this be happening? There's several possibilities. the virus has to recognize cleavage site. Maybe it's off target and it doesn't matter for the virus overall. That's probably the least interesting possibility. Traditionally, we've always thought of viral cleavage of cellular proteins as being a way to inactivate proteins that maybe form part of the immune response or antiviral response. So it's a way for the virus to switch off proteins that are detrimental to its replication. But the last option was actually that we're not switching off these proteins. Instead, we're altering or modifying their function somehow. So this virus can use cleavage as a mechanism to alter the activity of cellular proteins it actually needs to benefit the virus. So how did we determine this? So we used this siRNA screen. Now, this isn't a perfect way to study cleavage because we're changing the overall abundance of a protein, not blocking cleavage. But if the cellular proteins being cleaved are inhibitory for the virus, we'd expect to see a boost in viral RNA levels. On the other hand, if the virus actually needs these proteins, we should see a decrease in the levels of virus replication we can see. So here was our experiment. We transfected cells with siRNA, allowed those cells to recover for 24 hours. We then infected with SARS-CoV-2, determined the level of knockdown we were seeing at this time, and allowed infection to proceed for 72 hours. And this was our data. So in red, we have proteins which are not significant, and in blue, which were t-test significant. And here is our mock here. So we have proteins we identified in our A549ACE2 cells, those in our Vero cells, and those from either cell line which had a poorer match to the consensus sequences that were known. And what we found was that quite a number of these cellular proteins showed a decrease in viral RNA copies that we would observe in infected cells. But actually, every target we looked at showed a reduction in the levels of infectious virus produced relative to our mock control here. And we do have additional controls in the paper where we show that actually if we do knock out known antiviral proteins, we do see an increase in viral titers. So this wasn't an artifact of our system. But it does actually say that actually we seem to be seeing the virus predominantly modifying the function of cellular proteins that it needs. So can we actually use this as a strategy to find new antivirals? So two of our substrates were the kinases Src kinase and myosin light chain kinase. There's an advantage to studying kinases, which is there's a number of inhibitors available to study these proteins. Sorafenib we used as a control, as this was previously published as diminishing SARS-CoV-2 infection. But we also found befetinib had a sub-micromolar IC50 against SARS-CoV-2. When we looked at myosin light chain kinase, we had a rather interesting finding that two different inhibitors, ML7 and MLCK inhibitor peptide 18, gave opposite results. And this was curious for us, as these enzymes have two different mechanisms of action on the protein. So it suggests we need to delve into quite how the cleavage products of myosin light chain kinase are functioning during infected cells and in infected cells to understand what is driving these different results. We did see several other Src inhibitors which had better IC50, but they did have increased issues with their cytotoxic 50% levels. It's more likely that these were cytostatic in this system. It's a known issue with the cell line that we were looking at. However, it does suggest that targeting the substrates of viral proteases has prospects for future host-directed antivirals. And this is a real gain for emerging viruses, Because if we know that families of viruses share the same host substrates, we may not know what a new virus from that family will look like, but if it uses those same substrates, inhibitors that target this mechanism can be effective against newly emerging viruses. So I'll summarize this first part of my talk. So I've told you about how we used N-termonomics to study proteolysis during SARS-CoV-2 infection. I've shown some data that we had cleavage sites identified in multiple viral proteins, and in this talk I've focused on some of the sites we identified in the spike glycoprotein. We found 14 entirely novel cellular protein substrates of the viral proteases, and these showed both cleavage recognition sites and temporal regulation consistent with cleavage by the viral proteases. And we showed that actually, counter to the prevailing model that viruses cleave cellular proteins to switch them off, SARS-CoV-2 appears to need all of the new proteins we identified, and that targeting these cellular proteins could be the strategy for developing new antivirals. Now, in the second part of my talk, I'm going to talk about some of our ongoing work on single-cell proteomics. So that's quite a change from performing N-termonomics. So what actually drives me to use single-cell proteomics? And why does my lab work on and develop these methods? So you've gathered by now that my lab predominantly studies RNA virus replication and virus-host interaction. But in cell line models like those you saw, virus infection, even with a clonal virus population, can be highly variable at single cell level. We have an example here. We have Vero cells that were infected with avian coronavirus, and we have either antibodies against a cellular protein caprin, against the nucleocapsid, and a merged image. And what you can see is that some cells show no viral infection. Some show massive amounts of viral antigen production, and others show a more intermediate phenotype. Now, this is a clonal cell line and infected with a clonal virus population. So what is driving this? And why is it interesting? So for me, one of the reasons it is interesting, and it's interesting at the protein level, is because, again, post-translational modifications are playing a crucial role in virus replication. In the first part of my talk, I told you that polyprotein cleavage was a very conserved mechanism in virus replication. Here I'm swapping from coronaviruses to a simpler virus, norovirus. You may have had the unfortunate experience of the human version of this virus. In the lab, we tend to play with mouse norovirus, which is happily harmless to humans, so we're not going to give this to ourselves. And it's very similar to the human version of the virus. It just has this additional immunomodulatory protein, VF1, that is expressed from an overlapping reading frame. Once again, the bit of this virus I care about is the polyprotein, which is cleaved by the internal NS6 protease into a range of fully and partially cleaved intermediates. Now, at single cell level, why should we care about that mixed picture of infection that we were seeing? Well, if we take a theoretical model, we have our uninfected cells here, we add some virus, we can have a few different outcomes. Our cells may stay uninfected, they've escaped infection. They may become infected but manage to recover. They may become persistently infected, or they may lyse and die, releasing a new virus. And really, the reason to care about single cell measurements during viral infection is to be able to work out what is driving these different branch points. So what drives cells to become infected or remain uninfected? What drives them towards either lysis or persistent infection, or persistent infection and recovery? And given the role of post-translational modifications in infection, the single-cell transcriptomics approach is not going to be able to give us the full picture of what is going on. So performing single-cell proteomics in this model is interesting, but what are our challenges? Well, the general challenge with single-cell proteomics is low sample abundance. You're talking in the range 50 to 500 picograms for a normal mammalian cell. We can only study a maximum of about 18 cells in a run. It's about 14 to 15, accounting for our carrier and reference channels. And there are not really kits available. Sample preparation and our mass spectrometry is not fully automatable or kit-based. So it is quite laborious and time-consuming to perform. So how do we actually prepare our samples? So during my postdoc with Nikolai, I worked with his lab on Scope 2, and you may be familiar with this approach for single-cell proteomics. So we sort by FACS cells into 384 well plates. These will contain our single cells. We'll also prepare bulk samples diluted down to 5 cell concentrations or 100 or 200 cell concentrations. This 5 cell sample we'll use as a reference for normalizing between our different TMT sets. Our carrier we use to improve peptide identification and also to help protect our very sample limited single cell samples from losses during downstream processing and LC-MS. So in the original Scope 2 protocol, cells were lysed by a freeze heat cycle. We then add trypsin and digest, and we barcode with tandem mass tags. Samples were then mixed, and we perform LC-MS. For those familiar with tandem mass tags, you'll know that at the precursor level, we see a single peak for each peptide. But upon fragmentation, we'll see reporter ions for our individual single cells. We'll see a reporter for our reference channel, which we can normalize to, as well as for our carrier channel. And our carrier channel, we largely discard for downstream quantification because you'll be aware of the issues with trying to perform with ratio compression with tandem mass tags. More recently, Nikolai's lab has released the new MPOP approach, which my lab's now adopted. This allows for preparation of single cells in low nanoliter droplets performed on a glass slide in a Sanomon instrument. So you dispense DMSO. You dispense your single cells into the DMSO, so they're lysed by DMSO. We can then add trypsin, TMT label, block our TMT, and then pool our TMT sets and dispense into a 384 well plate, ready to dry down and then inject into the mass spectrometer. So both these methods sound great. We're using tandem mass tags and standard mass spec commercially available reagents. So can we use this for some useful biology? So in the Scope 2 paper, we took a very simple system. We took U937 cells. These were either mock treated or treated with PMA, which results in differentiation from a monocyte-like to macrophage-like state. And we took these cells then at 72 hours post-infection. We were able to perform analysis of just under 1,500 cells, identifying about 3,000 proteins. Now, because we'd FACS sorted our cells, we knew up front which our monocyte cells were, shown here in red, and our macrophage cells here. But if we actually look for proteins known to be enriched in monocyte or macrophage lines, we saw this expected pattern as well. Now, you can read more on this in the Scope 2 paper, but I'm going to focus on some new data from my lab. So very recently, we've been starting to apply this to viral infection time courses. And we started off with perhaps a slightly too complex model system, where we infected or mock-infected cells, and we looked at these cells at 6, 8, and 10 hours post-infection. Now, it's important to note that we infected these cells with a level of virus we did not expect to result in 100% infection. And also, due to the variability in infection, we also expected these samples to somewhat blur into each other. And indeed, this is what we see. So here in blue, green, and purple, we have our infected cells. They're largely here. And if we perform clustering, then it actually makes this a bit clearer. We have our early time post-infection cells here, merging into our late time post-infection cells in cluster 2, with cluster 3 being our uninfected cells. So cells by PCA seem to progress from here into cluster 1 and then towards cluster 2. So they're doing kind of loop. Now, if we look for individual viral proteins, we can see in this top cluster, we can see increased expression of the polyprotein here, of the main capsid protein here, and of the VP2 protein here. And we can see these in the upper end of each of these PCA plots. Now, if we actually look at specific viral peptides and their expression, we actually identified peptides which corresponded to some of these viral cleavage sites. And it's when we look at these, highlighted at NS4 here, we start seeing some interesting patterns. So we can see that from early in infection, which is showing up on the right here, where we see low levels of viral protein expression, we then see an increase in the presence of this cleavage site here, a temporary increase in capsid protein expression, followed by a drop, and then most of these other proteins show increases. This is not something we've ever seen in our bulk proteomics data, because all of this is averaged out. In our single cell data, we're starting to be able to pick out new patterns, which may explain how the mechanism of how the replication processes are regulated at single cell level, because we don't have this bulk proteomics averaging effect. Now, we're interested in very specific peptides of interest. So we're shifting to the P-scope approach, which I was working on for part of my time in Nikolai's lab, Slavov's lab. And this uses MaxQuant to target particular peptides of interest for analysis during mass spec runs. So it's instrument control software. And to give an example, if we take our different viral peptides of interest, this is a standard sample. Blue, we don't detect. Yellow, we detect. And this is just a normal DDA analysis. If we, however, perform this by P-scope, you'll see that actually there's one peptide we still don't see in all samples. All our other peptides we're seeing in most of our samples, including some that we'd previously never detected just by a standard DDA approach. These are for particular modified peptides of interest, which is why we have so few on this plot. But if we expand this to all the viral peptides we identify, you can see this picture holds. Even where we're not able to identify in every replicate, we're seeing significant increases in how many samples we're able to identify peptides of interest in. So to summarize my lab's ongoing work on single cell proteomics, these methods allow analysis of thousands of proteins across thousands of cells. When we apply these approaches to viruses, we can identify post-translational modifications of interest. And these are occurring in distinct clusters and with patterns that actually appear to suggest mechanisms of viral regulation. And by applying targeted analysis during infection, we can significantly enhance our reproducibility of detection and our ability to detect post-translational modifications and specific peptides of interest. So especially the first part of my work was a huge collaborative effort during the pandemic, both with my lab, collaborators at the Institute Pasteur, Charlie Craig and the Verdi-N lab, Jan Gambin and Emma Sirecki at University of New South Wales, as well as at UOL. And the single cell work I started presenting at the end has been led by Stephen Holmes. Thank you all for listening, and I'll very happily take questions.
Thank you very much, Ed, for this nice talk. I will wait some questions in the chat here from the audience. I have some questions and several in fact, but I will ask some of them. How, in your experience, Ed, how many cells do you think we should start in this single cell analysis? For example, in the beginning of the project, we analyze a specific disease or maybe cells in the blood or cells in the tumor microenvironment. What is your advice about how can we start?
So when you're first trying to set up the method and you're just doing, say, 2 or 3 cell line comparisons, tens or 100 or 200 cells are going to be plenty to check your method's work. For more interesting examples, I tend to use power calculators that are designed for single-cell RNA-seq, and they're based around what is the smallest population of cells you want to be able to distinguish, and how many cells do you need to be able to see in that population for it to be something that you trust and believe. So I tend to use power calculators that have been developed for single-cell RNA-seq.
Thank you very much. And another question, if you can compare, for example, use multiplex samples and label-free, about the intensity, about how can we compare these results, and how could you suggest for a lab that is starting with single cell?
Yeah. So in my lab, to date, we've only really done multiplexed analysis. It took a while for them to arrive. We finally have MTRAC reagents. So we are very interested in doing plex DIA based multiplex DIA analysis using MTRAC. We're somewhat interested in label-free for very niche applications. As Nikolai's lab showed in the plex DIA paper, the data does actually align very well. You do get very good agreement. I think due to throughput, if I were, for most experiments, I would plan to use a scope type approach for the bulk of my data set. And then you validate the change that you see in a smaller subset of cells with either label-free or a plex DIA approach to get the more accurate quantitation.
Okay, thank you very much. We have a question from Daniela. Can you read there?
Yes, I can. So the question says, for P-scope, what criteria do you use to prioritize your peptides? So exactly as in the P-scope paper, we start by doing a DIA analysis of a sample that is more abundant than a single cell sample, but say it's 10 times the amount of material. So 2000 cell carrier type amount, to see what can we detect at that level and to get accurate retention times. Now, for my specific research here, the thing I really care about most are those modified peptides that represent the cleavage sites in viral proteins, either the uncleaved or the cleaved versions of these peptides. So P-scope has the advantage that basically you assign all the peptides to different groups based on how much you want to see them. So my top group would have those modified peptides. The tier below that would be other viral peptides for increased confidence. Below that would be cellular proteins that I'm particularly interested in. Perhaps I did a untargeted scope analysis and I found that these were changing in my different populations, or these come from bulk proteomic analysis, and I know that they change during infection. So they're proteins I know to be interesting. Beneath that, I would have 2 peptides for other cellular proteins, and I'd select the specific peptides based on their intensity and reproducibility of detection. Beneath that, you can have further tiers. And how pScope works is it will work through all the top tier. If it then has remaining cycle time, it will work through the next tier down, the next tier down, and so on. So you aren't wasting instrument time by having more tiers. Yeah. So I think some of the nicest data for this is on Nikolai's Plex DIA paper, where they show you can use both DIA-based and multiplex scope type experiments, and ultimately you're pulling out the same data in both. For any single cell experiment I do, as a control, I would always do proteomic analysis of the bulk samples. So for me, that's my infected and my mock samples, because I'd expect that if I take all my infected single cell data and I average that, and I take all my mock single cell data and average that, they should correlate with that bulk data. And that is what we see. For increased reproducibility, you have a few options. You have approaches like pScope, if you're going to go with TMT-based methods. You also have DIA-based approaches, and I'm a real fan of Plex DIA, and we're hoping to get that set up in the lab very soon.
Thiago, do you have any questions to add?
I have one question, actually. It was just a great talk, and you're just an awesome speaker. As a plant scientist, I'm always worried about the complexity of the plant cells. So do you have an experience on working together with plant scientists to unveil, the complexity of those cells? Because as far as I can notice, it is kind of mandatory to have an in vitro system in order to analyze the single cell proteome. Is it correct?
Yeah. Other labs are starting to look at more complex tissues and so on. I don't recall seeing any plant single cell data yet. I'm sure it's being worked on. I'd be very interested to see how it looks. But I think it's a matter of, if you have a way to disaggregate your sample so that you can use either a cell-in-one or a cell sorter, then you should be able to prepare your cells.
That's true. So it is kind of mandatory to reduce the complexity.
I think if you have methods that work for single cell RNA sets to break up your plant tissue into single cells that you can analyze, then hopefully they will transfer over to single cell proteomics.
All right. Thanks, Ed.
You're welcome.
And for the complex tumor microenvironment, for example, do you think is it a good idea to use FACS and then cell-in-one, because cell-in-one has limitations in terms of labeling? Have you ever used these two instruments in sequence?
Oh, FACS and then cell-in-one. I mean, I would generally perform the cell sorting in using the cell-in-one's own capabilities. Now, I believe there is an early application note from, I think it was the Meckler lab, where they actually used the cell-in-one to prepare samples in dishes. And it is doable. It's there are probably cheaper bits of equipment to perform plate-based sample preparation, but it can be done. I think it's certainly at the moment there isn't a good way to integrate the CellM1 with a means to retain some of the spatial information of your sample. So currently we have to disaggregate our sample. We get a mix where we've lost all that spatial information. So currently there isn't any published workflow which gets around that, or one that I'm aware of.
Thank you very much. There is another question from Reinaldo.
Yeah. So question is, in N-terminomics, do you expect smaller peptides? If so, do you need changes in LC-MS parameters or use of alternative enzymes? In N-terminomic analysis, particularly with the way we do it, we label at the protein level pre-digestion. So actually we get longer peptides because trypsin will not cleave at a TMT labeled lysine. It will cleave at the arginines, so effectively trypsin behaves like ArgC. So you get slightly larger peptides often. Those peptides are going to determine which cleavage sites you can see. So use of alternative enzymes is a definite consideration, and one of your countrymen in my lab has been doing a lot of hard work getting that all working very nicely for us. So nowadays we tend to use multiple proteolytic enzymes to get better coverage of different cleavage sites.
Thank you, Ed.
You're welcome.
I think you do not have more questions. So I'd like to thank you very much. Did I have check?
Oh, that's okay. I can ask later.
No, you can ask here.
I was just wondering, because regarding the first part of your talk, Ed, so you're basically identifying novel cleavage sites, right?
Yeah.
And can you differentiate like endogenous peptides from those novel peptides that you're generating by?
So for us, our primary differentiation is based on quantification. So it's because we have those time point samples, we identify all these different peptides, but we're limiting our downstream interest to ones that are showing changes upon infection. So the original Hunter method blocks lysine N-termini with formaldehyde. We wanted a method that would allow us to multiplex and combine all our samples early on, because I prefer not to do any sort of PTM enrichment where I have to prepare samples in parallel. Much better if I can combine early on and prepare a single sample and control for any technical variation in the enrichment.
All right, Ed. Thank you.
Thanks a lot.
So thank you very much, Ed, for the nice presentation and for sharing with us your experience and your results. Thank you very much.
Thank you very much for the invitation.
So bye-bye.
Thanks a lot. Lovely meeting you both.
Thank you.
Thank you. Okay.